We see these leaves every day in our environment. But do you have ever wondered about what happened inside of them? These leaves are also a living creature, just like us and these chickens. As a living creature, these leaves also has a means by which they feed themselves. A specialized metabolism process called photosynthesis happened in the chloroplast, specifically in the membrane of thylakoid. For photosynthesis to happen, the presence of light, carbon dioxide, and water are critical. This time, we are going to simulate a process of photosynthesis, a simple one, and we are going to compare a photosynthesis with and without the presence of light. First, you need to find a green leaf, and then you cut it into several pieces that are roughly the same size. Next, you need to prepare 2 clear cups. You fill each cup with approximately 300 milliliters of water. Then you add each cup with a drop of dish soap and also 1/8 teaspoon of baking soda. And then stir it and make sure you don't make any bubbles. Next, you put 10 pieces of the leaves into the barrel of the syringe, and then you put back the plunger into the syringe and you push until there's only a little bit space left. Put the tip of the syringe into the water mixture, and then you pull the plunger until the water hits the 3 milliliters mark. And then you push again the plunger to push the air out from the barrel of the syringe. You put your hand on the tip of the syringe and then you pull the plunger again to create a vacuum space, and then you leave it there for 10 to 15 seconds whilst shaking it. Do it until all the leaves fall down into the bottom. And then you remove the plunger from the syringe and pour the water and the leaves into an empty cup. And then you pour the water mixture into this cup until approximately 3 centimeters high. You put the cup in a place with sufficient lighting, and then you need to observe at what time half of the leaves are floating in the top of the water. And then you need to redo all of the steps again, and you put the cup, this time on a dark room. These are the results that obtained from the experiment. It shows that the cup that was put in the room with sufficient lighting has high rate of photosynthesis indicated by the time that half of the leaves took to travel into the surface of the water. And then compare it with the cup that was put in the dark. Initially, there were 3 leaves that immediately stationed at the surface of the water. But during the 15 minutes of observation, there were not a single other leaves that make its way into the surface. These 3 leaves finally sunk back into the bottom at 17 minutes mark. This means there is absolutely no photosynthesis that happened without the presence of visible light. The addition of dish soap was meant to lower the water tension and enables air bubbles to perform in the water as the result of oxygen production in photosynthesis. This bubble would at some point bring the leaves up into the surface of the water. Next up, we are about to discover the effect of temperature to the rate of photosynthesis. Basically, do the same setup as previous, but this time we'll vary the water temperature instead. There are 4 variations of temperature, namely 20 degrees Celsius, 25 degrees Celsius, 30 degrees Celsius, and 40 degrees Celsius. And these are the results. From the results shown, we can conclude that 25 degrees Celsius was the best temperature for the photosynthesis with the lowest half time, followed by 30 degrees Celsius by a close margin, and at 20 degrees Celsius and 40 degrees Celsius respectively with quite a big margin. The leaf that has been used come from a tropical vegetation Ficus septica Burm F. This plant is a type of sea tree plant, which can be observed by this chart, have the highest rate of photosynthesis at around 25 degrees Celsius. This chart shows photosynthesis rates of Ficus insipida, which fall under the same genus as Ficus septica, also shown to top at 25 degrees Celsius, and quite close to rate observed at 20 and 30 degrees Celsius. Both publications shows that the rate of photosynthesis take a steep decline at 40 degrees Celsius. This quite literally approves our findings. At biochemical level, net photosynthetic carbon assimilation is largely determined by Rubisco efficiency and activation and ribulose bisphosphate or RUBP regeneration. The predominant determinant varies with chloroplastic concentration of carbon dioxide, RUBP regeneration limits of carbon assimilation at elevated level of carbon dioxide. But Rubisco performance limits carbon assimilation at ambient and subambient level of carbon dioxide. The photosynthetic saturation and compensation photon flux densities generally increased with increasing temperature, but enzyme degradation at elevated temperature can impede the function of photosystem II. Decreased electron transport rates inhibit Rubisco activase or RCA and decrease chlorophyll content. Elevated temperature can also induce membrane permeability, leading to direct damage of the chlorophyllized thylakoid membranes, which further inhibits light harvesting, electron transport rates, and ATP generation. However, thermal lability of enzymes directly involved in carbon assimilations remains in a major cause of photosynthetic inhibition of C3 and C4 crops grown under elevated temperatures.